have come to give updates tonight that we're not going to have time for a live Q&A, but it's okay. Ask your questions, put them all in the Q&A box or in the chat. We're going to take them all and we're going to make sure that we get them answered for you, and we'll get them out to everyone in a PDF afterwards, just because it's going to be a long session because we have so much exciting things happening in the community. We should all be really excited to hear what we're going to hear tonight. I also, before we kick it off to the industry session, I want to remind you all that you have a chance to help the ASF out right now. If you can text ASF to 90412 and start giving feedback. What this is a program that if you give feedback, if you give 75 pieces of feedback, they will donate $125 to the Angelman Syndrome Foundation. We have a chance to get up to $50,000, which would be huge for our organization. It could cover 5 clinics for a year. It could cover a grant for research. It covers 50 families getting into the ASF Family Fund, and all you have to do is text ASF to 90412 and give feedback. So please join us in that. I want to thank you all for being here tonight. I know that as a parent to Jackson, who you just saw, who's off to bed, that this is really an exciting time for the community. There are so many pharmaceutical companies in the space. I think there's over 23 now. We're going to hear from all of the industry partners tonight that are doing any work within trials, preclinical, and hear the updates on what they're doing. So hopefully this will get you excited. Like I said, go ahead and enter in all those questions in the Q&A box, and we will make sure to get those answered. So without any other pause, because I know you guys are all here to hear from the industry. Have fun. We're going to kick it off, the 2021 ASF Virtual Conference Industry Update.
Hello to the ASF community and everyone watching online. I'm Becky Crean, Executive Director of Clinical Development at Ionis Pharmaceuticals. And on behalf of myself and the rest of the Ionis team, we'd like to thank you for listening today and taking time out of your busy lives to hear about our Angelman program. We had sincerely hoped that by this time, we'd all be meeting in person, but better to be safe and healthy. So we look forward to seeing you all next year. First, I want to thank the Angelman Syndrome Foundation for inviting us to be here. We're thrilled to be talking to you today about our program and the advances we've made since we spoke with you last year. So during this presentation, I'll introduce you to Ionis and our experience with developing ASOs. I'll give you a brief review of what we've accomplished so far in the program and where we are today. And then I'll give you an update on our clinical study, when it may start, and describe some of the details of the study. So there's a large group of researchers that are dedicated to our Angelman drug development program at Ionis. You may have met some of them before or heard them give presentations here at this conference, but these are just a few of the core team members that have been working on this program for many, many years. I'm Becky Crean, project team lead for our Angelman program. There's Frank Bennett, Chief Scientific Officer and Neurology Franchise Head. Eugene Schneider is our Executive Vice President and Head of Clinical Development. Christina Boyer is Vice President of Patient-Centric Drug Development. We have Matt Buck, Vice President of our Regulatory Department, Frank Rigo, Vice President of Functional Genomics, and Payman Jaffarnejad, who's Executive Director of Neurology Research. Now, there's a lot of other people on our team, too many to have pictures here, but they're all actively working as we speak on helping us to get a treatment to the Angelman community. So many of you may be familiar with the company, but I realize there may be new people attending to the conference since it is virtual and accessible across the globe. So I'll spend just a few minutes introducing you to Ionis and then speak specifically to our Angelman program. So as background, Ionis was founded in 1989 with a single mission. We wanted to turn antisense oligonucleotides, it's a mouthful, we call them ASOs, into a therapy. And so since the discovery of ASOs, 100% of our focus has been on developing and advancing this ASO technology. And we're committed to this technology in a way that might seem stubborn to some. A lot of other companies have tried it and said it would never work. It's now working. We've had some great successes along the way, and now more companies are coming in with similar technology. Ionis, though, is the first company to really apply this technology to treat diseases in a number of therapeutic areas, and one of the first to apply it to neurological diseases. We developed Spinraza, which you're familiar with, to treat spinal muscular atrophy, also Tegsedi to treat polyneuropathy, and others. And we have a pipeline full of ASOs in development and many in the clinic for the treatment of a variety of neurological diseases like Parkinson's, Alzheimer's, ALS, Angelman syndrome, and a variety of others. We have about 500 employees and counting. Maybe half of those are in research. We have a large core research group that doesn't work on any particular disease. They only work on understanding the mechanism of ASOs and how to deliver them and how to make them better. The other half of the company works on disease-specific research, both in preclinical and clinical research. So the vision was to be a leader in the biotech industry in developing ASOs and to treat a range of disorders, and we continue to be laser focused on that mission. So that was a little bit about Ionis and what we do. Now let's talk about Angelman syndrome. And I know for this audience, I don't really need to introduce you to Angelman syndrome and the molecular mechanics, but I thought I'd talk about it as a way to introduce you to how our drugs work. So as you know, Angelman syndrome is due to a deletion or a mutation of the maternal copy of the UBE3A gene. And why this matters is because the paternal copy is silenced in neurons in the brain by this long antisense transcript you see here. And if you have a mutation or a deletion of UBE3A in the mother's copy, you no longer produce the UBE3A protein in the brain. So our approach using ASOs, which are small RNA or DNA analogs that are designed to bind to the RNA, those are designed to bind to this long antisense transcript that silences the paternal copy of UBE3A. So our ASO unsilences that paternal antisense transcript and thus increases UBE3A production in the brain. And we started working on Angelman syndrome quite some time ago, around 2012, with Dr. Art Baudet at Baylor College of Medicine and his team. And through a number of years, the teams were able to validate this ASO approach and validated that it could unsilence the transcript and reverse some aspects of the disease. We showed this in animal models. And so since then, we've been leveraging our 30 years of experience in antisense technology and advancing our Angelman program. Our due diligence in selecting a candidate for clinical development is quite a methodical process. And we start with an extensive screening process, screening over 3,000 ASOs. And from there, we start to identify several leads. And those leads go through a number of preclinical studies, mouse studies, cell culture studies, and ultimately non-human primate studies. So each process helped us select out the safest, most active and potent ASO to continue advancing. So as a result, we've had several lead candidates identified through this screening process and advanced several of them into toxicology testing. So as we sit here today, we and our partner Biogen continue to make progress in this program. Our research studies continue, and we remain 100% committed to delivering an ASO to the Angelman community. So where we are today with our program, I think most of you are aware, we announced last December, we were planning on initiating a clinical trial with our lead compound this year. So through the testing, we identified a lead drug that we will test in our first clinical trial that we're anticipating starting by the end of this year. So to get ready, we've already started initiating important study startup activities such as country and site selection for this first trial. I'll tell you more about that in a moment. One important aspect of ours and any drug development program is to make sure we have the tools to adequately measure how people respond to the drug. To do that, we're partnered with Roche and Biogen in a natural history study to collect cerebral spinal fluid or CSF samples in individuals with Angelman syndrome. Some of you may have already participated in this study, and your participation has helped us start to determine how much UBE3A is in the CSF of individuals with Angelman syndrome compared to those who don't have Angelman syndrome. and to identify important biomarkers that can help inform and also de-risk not only our program, but other programs as well. It's also helped us design our trial to be more efficient, which means less burden on the participants and their families. If you haven't participated, the study is still enrolling. I know Biogen will tell you a little bit more about it. So there's still an opportunity to contribute to this important study. You'll also receive a priority voucher that can be used in either the Roche or the Ionis clinical trial. So our goal at Ionis is to bring the best drug forward. We want to emphasize that we believe we have a compound with the best risk-benefit profile that will treat the broadest population in Angelman syndrome. And we are 100% committed to bringing the best possible drug forward to the community. So I want to take a moment. I know this is a busy slide, but just to describe the drug discovery process. I've been talking about it in a bit more detail, and here we are within it. I just described to you the very lengthy and methodical process that's involved in discovering an ASO. And it starts here with basic research that you see on the left side of your screen. It involves identifying the target for the drug, and in our case, identifying the gene. And we work on what approach we want to take for that target, what protein to focus on. In this case, it's UBE3A. Then in early discovery, we focus on optimizing our lead candidates, which I just talked to you about. Then we move into preclinical research where we test the drug in different animal species. This all has to be done before taking the drug into humans. And that's where we are today, where you see the today sign. We've completed all the discovery and preclinical work from our lead drug. We're filing our IND application. From there, we move into human clinical trials. And there are several phases in that process, which I'll talk about in a moment. But after many studies and collecting a lot of data on the drug, which shows that it's safe and hopefully effective, that it treats the disease, then you file your data with the FDA, which gets reviewed. If they determine that the drug is truly safe and that patients benefit from the drug, then you get FDA approval to make the drug available. From there, you may continue to do additional studies. These are post-marketing studies, sometimes called phase 4. Those are a way to collect additional safety data and to answer any questions about the drug the FDA may have had during their review. So while discovering a drug and how it works is long and involved, so is the process of determining if it's safe and effective in people. So clinical trials are the development phase of drug development. And there are several phases of clinical trials that you must advance through in order to determine if your drug is safe and effective. So there's 3 main phases of clinical trials, each with their own goals and objectives. The first you see on the left is phase 1. This is the first trial in humans. And the primary goal and objective of that trial is to provide safety information of the drug and also to determine how the drug distributes in the body. These are typically very small studies, at most 50 people, and are usually done in healthy volunteers. And usually only 1 dose of the drug is given to each volunteer. So from there, you move into phase 2. These are a little bit larger studies. They're typically done in patients, in the patient population, and usually anywhere from 50 to 100 patients. The goal of the phase 2 study is to demonstrate that the drug is engaging the target and to provide at least preliminary evidence that the drug provides some clinical benefit. They usually involve giving multiple doses of the drug over a period of time, typically 3 months, maybe up to a year. But in rare disease programs, phase 1 and 2 studies are typically combined, which is certainly the case in Angelman syndrome. Then there's phase 3 clinical trials. These are much larger studies that are designed to establish safety of the drug in large numbers of patients, in the hundreds to thousands of patients, and also to show evidence of clinical benefit. These are the trials that provide the information to the regulatory agencies needed to approve the drug. So since clinical studies are conducted in people, both healthy volunteers and patients, many of you may have an opportunity to participate in one. But how do you know if you should or shouldn't participate in a trial? And we recognize that it's an incredibly important decision. It's a big commitment, so it's important to discuss it with your family. It's important to educate yourself as much as you can about what it means to be in a clinical trial and what's expected of you and your family. The ASF and FAST have put together a great website noted here that has a lot of helpful and detailed information about what it means to participate in a trial. I would encourage you to check that out. It's also important to talk with your child's doctor to get more information about the drugs that are being studied in the trials and what would be expected of you in a particular trial if you did participate. If you did decide that participating in a trial was right for you and your family, there's an important step that must happen before you can enroll. And that is you have to give your informed consent to participate. That's the first step in this process and an important one. Before any study procedures can be done, you'll be told all about the study and be able to ask all the questions you may have so that you can make an informed decision to participate or not. If you do and you give informed consent, then your child can be screened. And if they're eligible to participate, then you can enroll. Those are the first steps in the process. But here are some other important terms to be aware of that describe those first steps of the process. After you've given informed consent, you'll start with a screening visit. These are visits to check if you have all the inclusion and none of the exclusion criteria for the trial. Inclusion criteria are those required factors that allow you to take part in the trial, and exclusion criteria are factors that prevent you from participating in a trial. And every study is different. They have their own inclusion, exclusion criteria, depending on the drug and the trial and what their questions they're trying to answer. So some of those factors may be a person's health, history, past treatment, age, or even where you live. If you're determined to have all of the inclusion and none of the exclusion criteria for the trial, then you're eligible to join. If you're eligible, then you'll be enrolled into the trial, which means you'll then follow all the clinical trial procedures that's laid out in the protocol that's associated with the trial. And that's different for every trial. So that's a little bit about the beginning process of participating in a trial. And while we're still finalizing the details of our clinical trial, I can share a little bit more about what we do know about this particular trial. As I mentioned earlier, we anticipate starting by the end of the year. The ASO, as you've heard before, will be administered with a lumbar puncture, and your child will be given anesthesia to make them sleep during the short time it takes to give the drug. I mentioned before that often with rare diseases, we combine phases for a clinical trial. So this one will be a combination of phase 1 and a phase 2 trial. And each participant will receive several doses of the ASO over a period of about 3 months. And everyone will receive drug. No one will get placebo in this trial. We'll enroll a broad age range in our first trial, including young kids and up to adults. But different age groups may be staggered and enter the study at different times. During the trial, you and your child will also be given tests to measure the things we know are most important to you, like talking, walking, sleeping, learning information. Some of these tests will be done in the clinic and some will be done at home. This will be a global study with sites not only in the United States, but all around the world. Our primary goal in this first study is to assess the safety of the drug and to make sure it can be tolerated. And we'll also measure important clinical changes in those areas like communication, cognition, motor, sleep, everyday functioning. I spoke earlier about a priority voucher, and Roche and Biogen have also talked about that in detail before. But if you participate in one of the natural history studies that we are collaborating on, you'll be given a priority voucher, which you can use in any of the drug trials, including this one. So this is a schematic of the visits and the timing in the trial. And as I mentioned, there'll be visits in the clinic when the drug will be administered and at home during the trial. So you'll complete some other tests that will capture meaningful and important activities of your child. You'll do that at home so that we can see how those change during the trial. So for this first trial, it'll be small, only about 45 participants, ranging in age anywhere from 2 years up to 50 years old. We'll enroll participants with either a deletion or a mutation in the UBE3A gene, and participants must be able to undergo sedation in order to receive the drug. So initially, the trial will last about 3 months with a follow-up period, but we'll amend the study to add a long-term extension so that we will administer the study drug for a much longer period of time once we have the toxicology data to support the extension. So this is a partial list of the countries that the trial will be run in, including the US, Israel, Italy, France, Canada, Australia. the UK, Argentina, Germany, and the Netherlands. It'll be a large global trial. This is just an overview that we have, that we know about today, and we'll continue to update the community as we confirm more details of the study and when sites begin to open for enrollment. And we'll work through the Angelman Foundations in order to get that information out to you. So with that, I want to express my sincere appreciation for your time today and for listening to our update. All of us at Ionis and Biogen are deeply committed to continue our work on behalf of the Angelman community, and we can't do it without you. So we thank you for your commitment to doing this together.
Good morning, good afternoon, and good evening, everyone. It's a pleasure to be part of the ASF Industry Update. My name is Brenda Vincenzi. I'm the Medical Director for the Angelman Syndrome Program at Roche. And today we spend around 10-15 minutes together. I'll give you an overview of the progress since ASF 2020, an update on Friesia's observational study, and an update on Tangelo, our phase 1 drug study. I will then introduce the PET study in healthy volunteers, and we spend a few words to describe the clinician and caregiver reported Angelman-specific measures. We've been working within the Angelman syndrome space for the past 7 years. We started back in 2015 by listening and learning from the Angelman syndrome community. In 2017, together with ABOM, we developed the disease concept model in Angelman. 2018-2019, in partnerships with Ionis and Biogen, we started the co-creation of Friesia's observational study. Recruitment for Frisia started in 2019, when we also started to prepare for the phase 1 Tangelo drug study. The first participants in Tangelo was enrolled in August 2020, and in 2021 we completed Frisia's. Frisia's, or Endpoint Enabling Study in Angelman Syndrome, is a non-drug study, so observational study, where the study sponsors are Roche and Genentech, and is co-founded with Biogen and Ionis. Frisia's has a particular decentralized design. As you can see, there are only 2 in-clinic visits. New digital approaches are included and is collaborative. The primary objectives of Frisia's are the feasibility and value of novel endpoints and biomarkers to inform a clinical trial design. We enrolled 40 participants with Angelman syndrome age 1 to 12, 10 adults with Angelman, and 20 controls, so individuals without Angelman syndrome, for comparison. Frisia's only ran in the US. Recruitment was completed in September last year, and the whole study was completed in May this year. Here you see a few numbers, the demographic data from the Frisia's participants. In particular, you see on the graph on the left, in green, the number of enrolled participants in total, and in orange, the number of enrolled participants per month. And you see, if you look at the orange numbers on the graph, you see like a peak at the very beginning, the first few months, and then you see, unfortunately, where the COVID hit, not only Frisia's, but other, of course, clinical trial around the world, and you see like a decrease of the number of participants enrolled per month. But overall, we were able to complete the study, in this case, the enrollment, in September last year. Of course, a big thank you to the community, to families and participants, to all investigators, and the patient organization for the help in achieving this very important goal. In terms of gender, you see that we have 55% of males individuals enrolled and 45% of females. Here you see in orange the Angelman syndrome individuals, and in gray the controls. And you see the age of individuals, and you see the overall, if you look on the right side, the majority of individuals are with Angelman syndrome are between the age of 1 and 12, and a few individuals are adults. Here you see the demographic data from the Friesios participants. As expected, we see a majority of deletions, both class 1 and class 2, followed by UPD and UB3A mutation. Things to be aware of. Friesios is a non-drug study in collaboration with Roche, Ionis, and Biogen. These are separate companies that are working on drug studies for Angelman syndrome. So Roche, we are working independently on our Roche drug study called Tangelo. Ionis and Biogen are working together on their Ionis and Biogen drug study. Eligibility for any current and future drug study will be based on specific criteria. Participation in Friesios does not guarantee automatic eligibility for any current or future drug study sponsored by either Roche or Ionis and Biogen. Once again, a big thank you to everyone involved in Friesios, all families and individuals with Angelman, all patient organizations, all trial site teams and study coordinators, academic collaborators, and study investigators. Tangelo, as you know, is our drug study, is a phase 1 drug study, is an open label, meaning that everyone enrolled receives the drug. There is no placebo involved. It's a multi-center study to investigate safety, tolerability, and pharmacokinetics and pharmacodynamics of UB3A LNA in participants with Angelman syndrome. We aim to enroll up to 66 individuals with Angelman, both males and females, age 1 to 12. Genotypes included are mutations and deletions. And locations, we have 4 countries and a total of 12 sites. Study duration is approximately 58 weeks. And here you see the Tangelo study design. Tangelo has an open label design, meaning that all participants receive the study medication. It's a multi-center study with adaptive, multiple ascending intra-participant dose escalation. The drug is administered through lumbar puncture. The primary objectives of Tangelo are safety and tolerability and pharmacokinetics. Exploratory are efficacy and pharmacodynamics. So you see there is up to 6-week screening period. After the screening period, the participant will enter an 8-week treatment period and will receive up to 3 escalating doses of the drug. And this will be followed by a follow-up period, safety period, for a total of up to 58 weeks. And here you see more in details the Tangelo study design with the first participants that was enrolled in August 2020 in cohort A1. So as you can see, there are 2 cohorts. Cohorts A in yellow are the 5 to 12 years old individuals with Angelman. Cohort B are the 1 to 4 years old individuals with Angelman syndrome. So we, in August last year, we started with cohort A1. We started with very low doses and we cautiously escalated the dose. At the end of cohort A1, we reviewed all the safety data from the small number of individuals enrolled in cohort A1. We have internal experts and ad hoc external experts that we can consult. We decided there were no safety concerns, so we proceeded with cohort A2 and B1. Once again, we started with very low doses, and also the escalation was, of course, very carefully done. At the end of cohort A2 and B1, we reviewed the safety data. There were no safety concerns, and we proceeded with cohort A3 and B2. And that's exactly where we are right now. All these 5 cohorts are fully enrolled. We are waiting for the totality of the data from cohort A3 and B2 in order to assess the safety data and eventually to proceed with the next 2 cohorts, B3 and A4. So far, in the first 5 cohorts, we have enrolled 32 participants with no observed dose-limiting adverse events, and all participants are doing well. No dropouts, so all families that decided to enroll in Tangelo are still enrolled. One thing that I like to remind the community is that the primary objectives of Tangelo are safety, as I described, as safety, tolerability, and pharmacokinetics. And efficacy, like pharmacodynamics, are only exploratory. So any efficacy data that will be generated, of course, especially from the first few cohorts, they will have to be carefully interpreted. And there are many reasons for that. So one reason is that we enrolled a small numbers of individuals. The second reason is that because there is a safety and tolerability study, we started with very low doses. And even if we escalated, the doses were still low. The third reason is that this is an open label, so there is no control, there's no placebo arm. And then the last reason is that unfortunately the endpoints available to measure changes in the Angelman syndrome population during a clinical trial are still not established. And now we change the topic. I'd like to introduce the PET study in healthy adults. This study represents a pioneering work that will inform not only our study, our program, but other LNA and antisense oligonucleotide programs. So what is a PET? PET stands for position emission tomography and is a medical and research imaging technique. And how does it work? It uses a radiolabeled UB3LNA, in this case, injected via lumbar puncture. After the radiolabeled UB3LNA is injected, the PET is performed. As you can see, the individual enters the PET scan. And we then look at the images generated to see where the UB3LNA distributes in the CNS, in the central nervous system. When did we plan this study and when did we start it? So Roche started to plan this study more than 2 years ago. We work on the protocol, we discussed with health authority until we had, of course, the protocol approved. So we started enrollment a few months ago. Why do we perform this PET study in healthy adult volunteers? There are several reasons. One is to better understand the distribution of what we call the biodistribution of our UB3LNA in the central nervous system in humans following intrathecal administration. To confirm the intrathecal injection protocol delivers the UB3LNA, the drug, to the brain at desired concentrations. To explore the impact of different intrathecal injection protocol parameters. And this allows refinement of the prediction of the UB3LNA effect on the UB3A protein in patients, which hasn't been measurable so far. As I mentioned, this represents a pioneering work that will inform several programs. The last topic is the development of Angelman-specific clinician and caregiver-reported measure to support holistic, patient-centered drug development. Measures assessing key symptoms and functional impairments of Angelman syndrome are needed to support therapy development. FDA guidelines highlight the importance of patient-centered drug development and outline a standard process to develop such measure. 1, to understand the condition. 2, to conceptualize the clinical benefits. 3, to select, develop, or modify outcome measures. We follow best practice FDA guidance to develop Angelman-specific measures for use in clinical trials. Robust qualitative methods were employed to elicit input from expert clinicians, patient advocates, and caregivers at multiple time points, as you can see along these gray arrows. First of all, we developed the Angelman syndrome conceptual model. We drafted the Angelman-specific measures. We interviewed clinicians and patient advocates to discuss these draft measures. We refine based on feedback. We refine the draft measures. We re-interviewed the clinician and patient advocates, and we interview caregivers to discuss these refined measures. So we came up with the Angelman-specific measures finalized or version 2. So these scales, so Angelman syndrome specific, clinician and caregiver reported, they capture the most salient concept of importance in Angelman syndrome. On the left side, you see the SAS-CGI, or Symptoms of Angelman-Specific Clinician Global Impression. This is completed by clinicians following interaction with the patients and caregivers. On the right side, in green, you see the CAS, or the Caregiver-Reported Angelman Syndrome Scale. This is completed by caregivers based on their own impressions. The items included in both scales are daily living self-care, seizures, sleep problems, maladaptive behaviors, communication, motor skills, cognitive impairment, plus one item that assesses the overall Angelman syndrome symptoms and one item that assesses the meaningfulness of any change. In conclusion, these novel measures, SAS-CGI and CAS, are designed to capture the most challenging Angelman syndrome symptoms needed treatment. Development following FDA guidelines, collaborating with Angelman syndrome caregivers, advocates, and clinicians resulted in content valid, robust measures. These scales are incorporated into Angelman syndrome clinical studies, which will allow evaluation of what we call the characteristics, the psychometric properties of the scales, and determine whether further refinements are necessary. This was my last slide. Big thank you to everyone for listening. Looking forward to your questions.
Hi, I'm Scott Sturma, Chief Medical Officer at Genetics. I'm going to talk to you today about GTX-102, an antisense oligonucleotide for Angelman syndrome. The clinical trial is also known as KICK-AS, knockdown UB3A antisense in kids with Angelman syndrome. Some disclosures here. GTX-102 is an investigational drug and is not approved by any regulatory authority. The data presented are interim data and do not provide definitive conclusions on efficacy or safety. Substantially more work is required to prove whether or not GTX-102 is safe and effective. Antisense oligonucleotides have been around for several decades. First drug was approved back in 1998. In the last 10 years, a number of drugs have been approved and a wide variety of indications. GTX-102's antisense oligonucleotide is a single strand of synthetic DNA and RNA. It targets RNA and alters RNA expression, so either increases or decreases protein expression. And this is a schematic of GTX-102. Now, GTX-102 inhibits our endogenous antisense that turns off the paternal gene. So if you look in the upper right corner here, you can see that the maternal gene in a neurotypic is on and produces a UB3A protein. Your body produces an antisense transcript that turns off the paternal gene. In AS, in the middle column here, you can see a middle panel. You can see that there's a deletion mutation. There's something that interferes with the maternal production of UB3A. The paternal gene is still turned off. What we're doing with the antisense oligonucleotide GTX-102 is you turn on the paternal gene. And so you interfere with the antisense transcript here that inhibits or silences that gene. And now you're turning it on and you get UB3A protein production. Now, this is an overview of the clinical trial. It's a phase 1 study, first in human study. There are 4 monthly doses. The dose escalation goes through multiple cohorts or groups. They all start at different starting doses and escalate within each group. There's no placebo or control group, and GTX-102 is given intrathecally by a lumbar puncture. We start with screening 4 weeks, then treatment over 4 monthly doses, and then follow-up for 6 weeks. The primary objective of the study is understand the safety of the molecule. Secondary is to understand the pharmacokinetics. That's basically measuring the drug in the blood and cerebrospinal fluid. And then the exploratory are looking at multiple functional domains of AS: communication, sleep, behavior, gross motor, fine motor, and seizures. The key inclusion criteria, and the inclusion criteria are based on a number of factors. We want a homogeneous population, so we can see if there are any changes. We want to make sure that there's no potential safety issues. We know that ASOs as a class can have some impact on the liver and kidney. So we want to make sure that there aren't any confounding medical conditions that could potentially confound safety. So first off, we have deletion, requirement for deletion patients, age 4 to 17 years of age, stable seizure control. They can have seizures. As long as they're on stable medication, a stable number of seizures, that's perfectly acceptable for enrollment into the trial. Normal kidney and liver function, and able to tolerate anesthesia. Key exclusion criteria: any change in meds or diets to treat AS symptoms in the prior 3 months, sleeping aids, supplements, ketogenic diet. We just want things to be stable prior to coming into the study and during the study. Inability to ambulate independently or with an assistive device or caregiver handhold. That's because we're looking at gross motor function, and we want to see if there's changes in walking. Any bleeding or platelet disorder, and would require intubation. Now, the exploratory endpoints measure multiple domains because we don't know which test measures the clinical change the best. And we don't know which of these parameters will change. Maybe it takes longer to have an impact on communication, for example, than it would on walking. So we're looking at a wide variety of functional domains. So in sleep, we have Actigraph, which is a wearable device. They wear it on their ankle or on their waist. And the child goes to sleep, and we see how often they wake up and how long they stay asleep. There's a diary that the parent completes, and then EEG. The EEG is a 14-hour recording, starts in the late afternoon, during the night, and when they wake up. So we get both wake and sleep. Seizures, EEG, as I've just mentioned, and then a diary. The ORCA is developed by out of Duke, and it looks at pragmatic, expressive, and receptive communication, as does a Bayley and Vineland. So we have 3 measures looking at communication. Cognition is a Bayley and the Vineland, behavior is the Vineland, and motor, we have the Active Myo, which is a device that's placed on each ankle. It's worn for 3 weeks, then it's worn again 3 weeks at the end of the study. And so we look at stride length, speed, number of falls. Bayley and Vineland also look at that. Now, because we don't know how these measures will change, and they may not be sensitive enough, or they may not be getting at the parameters that are most important, we also have open-ended parent interviews. This is done by a third party. They call the parents and talk to them at the beginning of the trial and at the end to see if what changes have been good or bad in their child. We also convened a caregiver advisory board. We had 7 caregivers provide thoughtful advice on the protocol and study conduct. Assessments, visit schedule procedures, family needs, how best to convey the requirements to the study, what's manageable for patients and families, and what can we do to reduce the burden and the stress of the study. Because in phase 1, there are a lot of visits, there are a lot of tests, and so those are things that we have to balance for the individuals and the families. And the high-level conclusions, I'm just summarizing it. Fact sheet for the family, information beyond the informed consent, details, minimize unnecessary procedures to the extent that we can. Multiple blood draws can cause anxiety for the entire family. Wearable monitors on the wrist are not practical, and so the recommendation was on the ankles or the waist. And we were able to use it on the ankles in all these children, the first 5 children that were treated, and they all tolerated it actually fairly well. Comfort items, helpful during the visit, for example, age-appropriate toys, Netflix cards, snacks, weighted blankets. Travel concierge service, this helps organize the visits to the clinic. Portable bed enclosures to be available at the hotels if travel is needed. And numerous other items were incorporated into the study plans, trying to reduce the stress and the burdens on the individual as well as the family. Now, the doses administered. We were able to enroll 5 individuals under the original protocol. The amended protocol will be different than the first 5 patients. And these first 5 patients were treated at Rush University by Dr. Barry Kravis because they were able to get started before the COVID pandemic. Now you'll note that there's a 10-fold difference between the lowest and the highest doses tested. We had 3 groups, 3 cohorts. So in cohort 1, we started at 3.3 milligrams and ran up to 36 milligrams. We had 2 individuals enrolled, a 10-year-old boy and a 5-year-old girl. They got 3 milligrams first, then a month later they got 10 milligrams. And then 2 weeks later, we had an independent data safety monitoring board. It was composed of 3 external experts. One was a pediatric neurologist who was expert in AS, a pediatric anesthesiologist, and a statistician. They reviewed the data and said, yes, it's okay for these patients to continue dosing and for the next cohort to start dosing. So in the second cohort, we had a 12-year-old boy and a 15-year-old girl. They started at 10 milligrams. A month later, they got 20 milligrams. 2 weeks later, the Data Safety Monitoring Board reviewed the data and said, yes, it's okay to continue dosing, and it's okay to start cohort 3. And we enrolled a 5-year-old boy. He got a single dose of 20 milligrams. Unfortunately, patient number 2 in cohort 1, after her 36 milligram, got a serious adverse event, radiculopathy, and I'll talk about that more in a moment. And that's when we stopped dosing in all patients and stopped enrollment into the study. And you can see here the last row. These are the cumulative doses that the individuals received. They received anywhere from 20 to 105.3 milligrams. Now, we did see transient ataxia and fatigue. Ataxia is wobbly walking. A lot of these individuals have ataxia to start with, and this made it worse. It appeared to be dose related. As we went to the higher doses, we saw it more and more severe. And it was primarily observed at 20 milligrams and higher. And there was variable severity between the patients. It started 2 to 6 hours after the injection, and it lasted 1 to 3 days. Patients were back to normal when resolved and able to eat and interact normally during the symptom. Other mild events occurred like headache, urinary tract infection, and other typical childhood infections. The patients tolerated the lumbar punctures and the anesthesia well. The serious adverse event was the lower extremity weakness or radiculopathy that was seen in all 5 patients. It occurred 6 to 30 days after the last infusion. There was significant weakness in 3, and in 2, they were unable to walk. And this occurred in the 2 youngest individuals. 4, this occurred after the 36 milligram dose, and 1, it was after the second highest dose at 20 milligrams. Dosing was paused for all patients after the first case was observed. The most severe aspects of these symptoms lasted 2 to 4 weeks, and full resolution took between 19 and 126 days. Now, we did a clinical global impression of improvement at day 128. This was done by the principal investigator at the study, Dr. Barry Kravis. And you can see here the patients 1, 2, 3, 4, and 5. And this is looking at global, so the red with the purple, and then fine motors in green, gross motors in orange, the turquoise is communication, behavior in the gray, and sleep in the dark gray. And you can see here for patient 1, much improved, and there is much improvement in gross motor and communication, and minimal improvement in behavior. Cohort 2, or patient 2 rather, and patient 5 were the 2 youngest, both 5 years of age, and they were very much improved. But you also see that the orange was a gross motor. The CGI here, this assessment was done at day 128, and this is when they were in the midst of the most severe aspects of the radiculopathy or the weakness in the lower extremities. And so you could see that there was minimally worse or much worse in these individuals. We retested this after that symptoms, that adverse event recurred, and they were all back improved. In fact, patient 2, she was able to swim, learn to swim without floaties, was able to climb over curbs, and so marked improvement after that serious adverse event. Patient 5, it's interesting, you can see sleep was very much improved for this individual. He would wake up 3 to 5 hours a night and wake up the family and the parents. But a couple of weeks into the study, he would sleep through the night. So that was a significant improvement for that individual. Patient 1 didn't really have a lot of problems with sleep, so that's why we didn't see a lot of change there. But overall, you can see very much improved for all the patients on average, and you can see improvement in fine motor and communication and behavior and some sleep. So small number of patients. We need to study more patients over a longer period of time to see how sustained and how substantial these changes can be. We also looked at ORCA, and this was the observer-reported communication ability. This was completed by the parents. And you can see for each row is each of the patients, and baseline, day 120, and there is additional follow-up that's done. And there can be natural variation, but you really need a 5-point change for it to be significant, either up or down. And we saw that for patients 1, 2, and 3. So overall in communication, it seemed to be a real change. Again, these are early data, small numbers, but it's encouraging. I think that's what this data shows to us, that there's encouragement. There's other data in EEGs and Bailey's that we saw some changes that were encouraging, but again, small numbers, and we need more data over a longer period of time, a larger number of children to see to make definitive conclusions. At this point, these are some of the anecdotal observations from the parents. And I mentioned to you that one child learned to swim, was able to go up curbs and walk up a grassy hill. Another child, it was interesting, a couple of the children, they wouldn't really respond to the name, but after being on the drug for 1 or 2 or several months, they would then respond when their names were called. And patient 1, for example, would only play with his brothers for like 5 minutes, but during the course of the study, he would then play with them for an hour. 2 children were nonverbal, developed anywhere from 2 to 11 words. So there was definitely encouraging reports from the parents. Now, what we've learned from this study, GTX-102 is active at lower doses. Based on what we saw in non-human primates or monkeys, we thought we'd have to go to higher doses, but we were actually seeing some changes clinically at the lower doses. We did find that the higher doses were associated with the serious adverse event of leg weakness, and the SAE appears to be related to a localized effect at the site of injection on the lumbar sacral nerves in the lower back. So we've amended the protocol. We're going into lower doses, 3.3 and 5 milligrams, depending on the age. We're individualizing the dose titration. So you saw earlier, we went from 3.3 milligrams to 36. We're only going to go as high as 14, and that's going to take a year to get there if they need it. If the patient's having improvement, they won't get their dose increased. If they're not improving, it'll go up at much smaller increments, 3.3 to 5 to 7.5 to 10, etc. So those are the changes to avoid the serious adverse event of the leg weakness that was observed in the first 5 patients. In the UK and Canada, the clinical trial application was approved by the Medicines and Healthcare Products Regulatory Agency, the MHRA in the UK. That's the UK's equivalent to the US's a version of the FDA. The amended CTA was approved by the Health Canada. They approved the original protocol and then approved the changes, the amended one. So we'll have 2 groups of starting doses based on age. The younger children will get 3.3 milligrams, and the older children will get 5 milligrams. We'll do 4 monthly doses over the first 3 months, and then maintenance every 3 months. And as I mentioned earlier, we'll do dose titration. If they're not much improved or better at least 2 domains, functional domains by the CGI, then they can go very small increments every 2 to 3 months. So it'll be much slower and smaller dose increments. In the United States, we've provided all the same information to the UK and to Canada and to the FDA. And at this point, we're still waiting for the FDA. We have some ongoing discussions with them, and we'll update the community as soon as we get the outcome from the agency. For clinical trial information, all new sites are activated, and that contact information for each study site will be posted on the kickas.com website as well as clinicaltrials.gov. So as a parent, that's where you'll go. You'll contact the site directly, and it's the investigator at each site that will evaluate the individuals and see if they're appropriate for the clinical trial or not. Additionally, once this information is available, we'll provide it to ASF and to all the patient advocacy groups in the US, in Canada, and in the UK. So thank you very much. Good night.
Good evening, everyone. My name is Nancy Jones. I'm the Vice President of Clinical Development at Neuren Pharmaceuticals. And I wanted to thank the conference organizers for inviting us to participate in this session. And I'm very pleased to be able to give you an update on our clinical development program for NNZ-2591, which is our compound that's being developed as a potential treatment for Angelman syndrome. A little bit about us. Neuren is a biopharmaceutical company, and we are specifically focused on rare neurodevelopmental disorders. And we are a small international team, and all of our team members are passionate about being involved in the development of new treatments for individuals that have great unmet medical need. And as you can see from this table showing our pipeline, we have two compounds that are in development, both of which are synthetic analogs of important naturally occurring molecules in the brain. In Rett syndrome, for the compound trofinetide, we have a program that's actually advanced to phase 3 and is expected to have results in Q4 of this year. And you can see also that NNZ-2591 is the compound that's actually in development for Angelman syndrome. It's helpful, actually, I thought, to be reminded of what the overarching steps are in drug development, and I'll give you an update on where Neuren is with respect to the clinical development program for Angelman syndrome. As you know, you start with your preclinical studies, which are the studies of the drug that are done in the lab, in either cell populations or in animal models. Then you do clinical trials, and there are 3 main phases to the clinical trials. Phase 1 are the initial studies that you do to look at safety and initial dosing, and these are usually small studies that are done in healthy populations. Then there are the phase 2 studies, which are small to medium-sized studies that are done in the patient population. The aim of these studies is to look at safety, but you're also looking at your initial evidence of efficacy in these studies. And then there's phase 3, which are the larger studies, which are designed to confirm what you see in phase 2. The primary focus of these studies is efficacy, but you do still continue to look at safety to confirm what the side effects of the drug are. Once the clinical trials are completed, you can submit a new drug application, which then can be reviewed and would get you your approval to make the drug available to patients. So where we're at for Angelman syndrome is that we have completed the required studies that are necessary for submitting an investigational new drug application to the Food and Drug Administration, or the FDA, and we've also completed the animal model studies. Since our last update at the conference last year, we have completed a phase 1 study, looking at the safety and dose, and we are planning to commence a phase 2 study for Angelman syndrome this year, and we'll be submitting an IND application to the FDA in the summer. So what is NNZ-2591 and how does it work? So to understand this, it's helpful to review what the different types of cells are in your brain. So you have neurons and neuroglial cells. And the neurons that you see in the picture on the left here in orange are the main cells that connect and send signals between the different cells in your brain to basically implement all the functions that you do. And they send these signals by their axons, and then they receive signals on the dendrites, which are these sort of branchy parts here. There are also other cells in the brain that are supporting cells, and I'd like to highlight the microglia here, which are in brown. And these cells are important because they are the supporting cells for the neurons, and they help with the maintenance and of both the growth and management of the cells, but also the connections between the neurons. All of these cells also produce an essential growth factor, which is important for creating new cells. And one of those is insulin-like growth factor 1, or IGF-1. And when IGF-1 functions normally in your brain, it breaks up into smaller pieces. And these smaller pieces are called metabolites. There's one larger piece that you see in the picture here that works on the IGF cell receptor and only on the cell surface. But there are other metabolites that work in other parts of the cellular pathway. And one of those is called cyclic glycine proline. And cyclic glycine proline is responsible for homeostasis of IGF-1, which basically means it regulates how much IGF-1 is available, making sure there's not too little, not too much. And what NNZ-2591 is essentially is a synthetic analog of NNZ-2591. So what we mean by a synthetic analog means that it has the exact same chemical structure as cyclic glycine proline, except for it has one small modification. And what that means is that it can actually mimic the actions of the naturally occurring molecule. So it has the same type of ability to regulate IGF-1 levels. But it also has the small change that you make also makes it have better properties to use as medication. So this is a novel molecule, a novel medication. It is an oral formulation, orally available, and it's a liquid formulation. And it has properties that are good for medication, meaning that it is 100% bioavailable, which means all the drug is able to be used and absorbed. It crosses the blood-brain barrier, and it has good pharmacokinetic properties. And pharmacokinetic properties are basically how the drug is used and absorbed. So why is NNZ-2591 relevant for Angelman syndrome? How can it be helpful? We know from preclinical studies that NNZ-2591 corrects features that are common in disorders with synaptic dysfunction. So if you've got abnormal function of your brain cells that maintain the connections between neurons, those microglia that I showed you in the previous slide, you can get as a result of that impaired signaling between the cells. You can also see an increase in molecules that signal to have inflammation. And then you can see actually a decrease in the expression or the availability of IGF-1. So NNZ-2591 corrects all of these things. So it puts them back into normal levels. And that then restores the balance that helps with the formation, maintenance, and the repair of the synapses, the connections between the neurons. And all of these effects of NNZ-2591 are state-dependent, means it corrects the impairment, but it doesn't impact normal cells. And that's actually really important with respect to safety. In Angelman, we do know that you see these deficits in synaptic plasticity or synaptic connectivity. That's seen in the strength of the connections between the neurons, as well as abnormal morphology and density of those dendritic spines, those spines that receive the messages. So all of that impaired signaling then results in the sort of symptoms that are observed. And what we've seen in a mouse model is that NNZ-2591 is actually able to rescue the phenotype of the Angelman syndrome mouse, including seizures. So these are the results that I'm showing you here now. And so this is with 6 weeks of treatment for NNZ-2591. And what you're looking at in the picture for each of these figures is one of the behavioral tests that is done, basically to look at an analogous behavior that's analogous to what you would see in terms of a symptom in Angelman syndrome. And then in each of these pictures, the black or gray bars are the wild type mouse, or the mouse that has no impairment or no sort of genetic dysfunction. And then the blue is the Angelman syndrome mouse. And on the left-hand side is, in each of the cases, is a comparison of what they look like when you treat them with just saline solution, so no drug treatment at all. And you can see for each of the behaviors, the Angelman mouse has sort of impaired behavior compared to what's normally expected for the wild type mouse. But when you treat them, which is what we're showing on the bars on the right, you can see that in all cases with the blue bars that the behavior of the Angelman mouse is actually improved or restored to be similar to what the wild type is. And you'll also note that if you compare the gray bars and the black bars, is that the behavior of the wild type mouse is not affected. So there's no negative effect on the wild type mouse. So in sum, what the preclinical studies tell us is that it shows us that NNZ-2591 has an effect on brain cells that improve structure and functions of the neurons and levels of IGF-1. It shows us that NNZ-2591 can improve analogous impairments in an Angelman mouse model, and it gives us some indication of what types of symptoms may possibly change when going into the patient population. And from what we've seen in the mouse model study, there's some potential to improve impairments across a range of symptoms. So what we've seen so far in clinical studies has been in the phase 1 study, and this has actually been a study in healthy adults. So the focus of this study was safety and tolerability, as well as to look at pharmacokinetics. Again, as a reminder, that's how the drug is absorbed and distributed and eliminated by the body. And so this was a 2-part study. There was a single dose stage, which was to establish first the initial safety of the dose by looking at a treatment with just a single dose. And then the second part was a multiple ascending dose stage, which was 7 days of twice daily treatment. And the group was either getting NNC-2591 or a placebo. And there were 2 dose cohorts, so a higher dose cohort and a lower dose cohort. And overall, what we've seen was a good safety profile for the phase 1. The adverse events were all mild or moderate and resolved during the trial. There were no serious adverse events or no severe adverse events. And with respect to lab tests, vital signs, or cardiac tests, there was no clinically significant findings. And the most common adverse event was drowsiness. There was only one discontinuation, and that was after the first dose. And with respect to pharmacokinetics, what we see is a predictable profile. And so it's the type of profile you want to see with respect to how you expect a drug or medication to behave. So our next step is a phase 2 study for Angelman syndrome. It's going to be a phase 2 trial, which is going to commence later on this year. It'll be in a pediatric population, and it will be conducted in Australia, and there'll be multiple sites. It's 30 weeks of treatment, and that's open label, which means that everybody gets the active drugs, everybody gets NNC-2591. And as I mentioned earlier, it's a liquid formulation and oral dosing. The primary aim of the study is to look at safety and also, again, to look at the pharmacokinetics, so how the drug is absorbed and distributed throughout the body. But the study will also look at the clinical response to drug, and we'll be looking at a range of outcomes in symptoms that are important to individuals with Angelman. Communication, fine and gross motor function, behavior issues, GI, sleep, seizure, and then daily function, meaning the activities of daily living. And all of this will also inform the design of the subsequent registration trials for our phase 3 trials. So currently, we are preparing to submit the investigational drug application to the FDA this summer, and we're in preparations for the study start. So that involves identifying and qualifying your sites to participate in the study, preparing the ethics review, and this is something that's required by all participating centers and as well as the FDA before the research can start, and also getting the study drug supply ready for distribution. So where can you find out more about information about the study? So we will be announcing more details about the inclusion criteria and the study as we get closer to the study start. We do have updates. These are going to be posted on our website, neurenpharma.com. If you haven't already signed up, you can get alerts. So every time something new is posted, you can get an email alert. So this is under news and reports on our website, and then under news and reports, you can click email alerts. And then once the sites are announced, the contact information and information on the inclusion criteria is also going to be found at clinicaltrials.gov. So clinicaltrials.gov is a clearinghouse of all ongoing clinical trials. So to find our study, you can search by using keywords Neuren and Angelman. So I wanted to thank you very much for your attention, and I want to wish you and hope that you enjoy the rest of the conference.
If we discover something cool in science, we end up in a textbook somewhere that's obsolete in 10 years. If we discover something that helps a patient, it lives on in the patients having improved lives. That's the reason why we do what we do. I'm Stormy Chamberlain. I'm an associate professor at the University of Connecticut School of Medicine. I work in the Department of Genetics and Genome Sciences. My lab studies chromosome 15q imprinting disorders. And so right now our efforts are focused primarily on Angelman syndrome. For all of our chromosomes, we have one copy that was inherited from mom and one copy that was inherited from dad. It usually doesn't matter, they're both evenly used. For chromosome 15q, that's not the case. For Angelman syndrome, they're missing a protein called UBE3A in their brain cells. To visualize how it works, I like to imagine 2 train tracks for chromosome 15q. One track represents the paternal copy. The other track represents the maternal copy. At one end of the track, they each have a train ready to make UBE3A. When a brain cell is functioning appropriately, the maternal train travels along the track and makes UBE3A. But on the paternal track, the situation is different. Traveling in the opposite direction is another train called the UBE3A antisense. Eventually, it knocks the other train off the track, and this is why the paternal copy of UBE3A is inactive or shut off. But since the maternal copy is functioning, there isn't an issue in a normally functioning brain cell. However, for patients with Angelman syndrome, they're completely missing their maternal copy of the UBE3A gene. What we need is a way to activate the paternal copy of UBE3A to prevent the train making UBE3A from getting knocked off the track. So because we understand how it gets silenced, we can figure out how to reverse that process. Our solution is to add an shRNA that cuts and derails the antisense train, allowing the paternal train to continue making UBE3A. And so we're going to put back the protein, the exact protein that's missing in kids with Angelman syndrome. Our lab uses induced pluripotent stem cells to study Angelman syndrome. We can take a skin sample or a blood sample from an individual with Angelman syndrome, and we can convert those cells into stem cells. The reason we want those stem cells is stem cells can become any cell type in the human body. So we can convert the stem cell to a neuron. And so in that way, we can study neurons from individuals with Angelman syndrome and test therapeutics in them. They'll plate the neurons on a plastic dish, and in each well of this plastic dish, they will add an shRNA. The shRNA it's nothing more than a set of scissors. It's scissors that we can direct, though, to a very precise place. So maybe we'll have 4 different shRNAs at a couple of different doses. They leave them on those cells for up to a week, and then they'll harvest the RNA. And what we're looking for is the conditions where the antisense is reduced and UBE3A goes up, because that tells us that the shRNA is cutting and that it's turning on Dad's copy of UBE3A. The final way we'll do this is we'll try a really big screen where we stick in tens of thousands of sequences. We develop cell lines that let us do it more rapidly. We use CRISPR engineering to allow us to convert them to neurons faster. When we initially started this work, it took us 10 weeks to make a neuron in a dish that we could then test an Angelman therapeutic approach on. We now have it down to a week. That's amazing progress. My people are happy.
Stormy is a great collaborator, and just her talent, her research talent is tremendous. This relationship is really about her knowledge and us leveraging it towards making a therapeutic.
What drives Ovid are the patients. I've kind of watched them be really mindful and thoughtful about what the data and what the clinical trial results means to the patients.
At the end of the day, that's who we want to impact. There's no other reason for us to do what we do. We are dedicated as a company, as a team, as individuals to working with Stormy and the foundations.
We want to get this right. The kids are too wonderful to accept anything less.
I can't think of anything more important, to be honest. We will make this succeed. That's our mentality.
With science, it's not an absolute, it's what we know today. And so based on what we know today, we should be optimistic. I think the whole world should be watching Angelman syndrome from that perspective.
Hello, my name is Katie Hogan, and I am a member of the Patient Affairs team at Tasha Gene Therapies. Tasha is on a mission to find potential treatments for patients with monogenic diseases of the central nervous system. We are using proven approaches to build the gap between innovation and a treatment. Our team at Tasha has deep-rooted experience in drug development with some of the brightest minds in gene therapy across the globe. I would like to start with the story about our name, as it's central to who we are. When we formed the company, one of the first things we had to do was settle on a name. Our founder, R.A. Session, has a 9-year-old daughter who also happens to be a Texas history buff, and she suggested the name Tasha. In the Caddo Native American language, it means ally or friend. When translated, it also means Texas. But this resonated with us beyond Texas history. First, we have deep roots in relationships with the biomedical community in Texas. Our world-class partnership with UT Southwestern is an illustration of this. Second, we are developing lasting alliances with the rare disease community. And third, as a friend and ally to the many we aim to serve, the name Tasha really captures who we are. You may have heard this before. It's critically important for 3 groups to work together to bring therapies to patients with rare diseases. These groups are advocacy, research, and industry. Strong patient communities help advocate for patients and raise awareness of diseases, making sure they receive the attention they deserve. Ties to academic and clinical research centers ensure discovery and science move forward. Industry support provides the resources and expertise to navigate the regulatory and commercial pathway. For me, this partnership has never been more apparent as we're working hand in hand with UT Southwestern and patient groups every step of the way. We're truly embracing an integrated approach, one that will help get new medicines in the hands of patients in the fastest, most effective way possible. Our partnership with the Angelman Syndrome Foundation and the entire patient community is critical to the success of Tasha's Angelman Syndrome program. I'd like to start with some ways we are working with advocacy today and future ways you can work with us. You can help Tasha understand the unmet needs of your kids and your families. You can help us understand areas of educational need for families and clinicians about our gene therapy program. With this information, we can develop clinical trials based on patient and family insights. And these interactions can help bring awareness to our clinical trials and make enrollment more efficient. Together, we can help advance new therapies for Angelman syndrome. When you look at how Tasha is built, there are 3 foundational building blocks for developing gene therapies quickly and efficiently. The technology we are using has already proven its merits. AAV9 gene delivery, the way we deliver and precisely target the dysfunctional genes, has been used in Zolgensma, one of the first gene therapies approved in the US, which has already treated more than 600 patients with SMA worldwide. UT Southwestern is a gene therapy powerhouse. Our partners, Dr. Stephen Gray and Burge Maniason, are among the brightest minds in the industry. In a short amount of time, they have built one of the largest pediatric neurology residency programs in the world. Their research and ability to translate these discoveries into therapies that can be moved into the clinic is unparalleled, and their innovative way to administer medicines and treat patients is world-class. We partnered with the best. Finally, we are building off this technology and research with a key leadership team that has direct experience in bringing gene therapies to market. Many of us have already worked together in the past, including at Avexis, the same company that developed Zolgensma. The fundamental components of our approach to gene therapy development are an adeno-associated virus serotype 9, or AAV9 capsid, intrathecal delivery, and an efficient manufacturing process. We use an AAV9 capsid to deliver a therapeutic gene engineered to replace a mutated gene, enhance the expression of a silenced gene, or decrease expression of a gene, depending on the underlying biology of the specific disease. Through our partnership with UT Southwestern, we have access to a good manufacturing process, or GMP, compliant manufacturing suite that we use to produce AAV9. We also intend to establish our own commercial scale, GMP compliant manufacturing facility to meet demand in the event that our product candidates receive marketing approval. We use intrathecal administration, which involves direct delivery of our gene therapies to the cerebrospinal fluid, or CSF, to facilitate optimal biodistribution and cell transduction within the central nervous system, or CNS. Our flexible manufacturing processes allow us to produce our gene therapy product candidates efficiently at scale. Through our strategic partnership with UT Southwestern, we have access to a world-class team of scientists and cutting-edge technology to discover and develop gene therapies. The scientists include Dr. Burge Maniason. He is a pediatric neurologist with expertise in neurodegenerative diseases, neurodevelopmental disorders, and genetic forms of epilepsy. We also work with Dr. Stephen Gray. He administered the first AAV9-based therapy to patients via the intrathecal route. Through the partnership, we also have exclusive access to next-generation platform technologies. At Tasha, we aspire to be an ally in the Angelman community. We are driven by a relentless focus on discovering and developing novel gene therapies for devastating disorders of the central nervous system. We remain steadfast in our commitment to understanding the unique challenges faced by the communities we serve, keeping your best interests at heart as we seek to deliver impactful and life-changing therapies for Angelman syndrome. As I mentioned at the beginning of our presentation, Tasha means ally. Our team is guided by principles of collaboration, transparency, curiosity, empathy, and respect as we carry out our mission of being an ally to those in the Angelman syndrome community. The strength of this bond with our allies will enable us to succeed. Thank you for having me at the 2021 ASF Family Conference and Research Symposium. For more information, please visit www.tashagtx.com.
Hello, my name is Alan Ria. I'm Vice President of Development at PTC Therapeutics and a Global Program Lead for PTC's Gene Therapy Program in Angelman Syndrome. I'm excited to tell you about PTC Therapeutics and the current status of our program in Angelman Syndrome. PTC is a multinational commercial stage biopharmaceutical company. We were founded in 1998 by our CEO and founding scientist, Dr. Stuart Peltz. We have offices in over 40 countries, including offices in the US, Europe, South America, and other regions. Given our experience throughout these 20+ years, we're experts in the development of treatments for orphan diseases. And our broad pipeline spans preclinical to commercial stage products. Now, PTC is a company focused on applying scientific expertise, our expertise, to bring novel therapies to patients with rare disorders. And we do so with proven science. Now, many of the platforms and therapies you see here were internally discovered and developed. Now, others were brought in to complement our scientific roots. You can see our gene therapy programs are a key part of our research pipeline, with products in development for multiple rare diseases, including Angelman syndrome. Now, we're passionate about our purpose. We want to bring meaningful therapies, those that will provide clinical benefit to patients with rare diseases. And over its 20+ years, PTC Therapeutics has shown itself committed to patients and caregivers in the rare disease community. As part of that commitment, we at PTC have built a global patient advocacy and engagement group whose members collaborate very closely with all of our program teams to ensure that the patient caregiver voice is represented in all that we do. And in our approach to Angelman syndrome in our program, we have a number of important objectives, including active connection with patient advocacy groups, networks, and associations to really establish PTC as a committed partner within the Angelman community. And in terms of key efforts that we're working on, of course, we have efforts ongoing to provide the patient caregiver experience directly to the clinical team and broader PTC to inform our preclinical efforts and our clinical efforts. We want to support education and awareness opportunities and share information and insights between patient advocacy groups and PTC. And ultimately, our group wants to empower patients and caregivers to become advocates for their healthcare. Now I'd like to discuss PTC's gene therapy in Angelman syndrome. Now there are a number of considerations related to the delivery of gene therapy in Angelman syndrome. The approach that PTC is taking is delivery of our gene therapy is by packaging the gene in a AAV vector. Now that vector is noted on the left here in red. We selected a specific AA vector that is appropriate for the treatment of a disease of the central nervous system. The vector reaches the relevant cells and tissue types of the central nervous system, which we want to happen. We know that UB3A expression, or the loss of UB3A expression, a gene regulated by genomic imprinting, causes Angelman. Now we designed the inside package of our vector, what we call an expression cassette, and that's what you see here in that long component here in the middle, to include the UB3A transgene. Now the expression cassette also includes a promoter. Now that promoter drives the expression of the UB3A gene once it reaches that targeted cells and tissue types. And again, that's what we want to see in gene therapy. Another key consideration is how we deliver the gene therapy. We at PTC have thought a lot about this, and there are a number of different routes of administration that could reach the CNS. As Angelman syndrome is a disease with broad CNS impacts, meaning many areas of the brain and the central nervous system are impacted, we believe that the best approach is to use a route of administration that leads to broad distribution of the gene therapy throughout the central nervous system. Now here on this slide, we see common pathway for researching gene therapy to support its use in humans. Now first, pharmacology studies, which are often done in rodent models of the disease, evaluate disease improvement and provide initial information on safety. Sometimes at this stage, larger animals are used. Now once there's good evidence of activity and safety in these early animal studies, a program classically moves to conduct official toxicology studies that regulatory authorities like the ANSM or EMA or FDA require. And these provide evidence of safety before the regulatory authority allows the company or the program of gene therapy to start initiating the gene therapy in human trials. While this may seem like a long process, these are important steps that need to gather enough evidence to support dosing in humans. At this point in time, PTC has conducted our initial animal studies, and we're planning our toxicology program to allow us to ultimately start our clinical trials. Now here I'd like to share with you some data from our early animal studies treated with our gene therapy. Now we treated Angelman syndrome mice and rats with our gene therapy, what I'll refer to as PTC-AS, and saw some nice evidence of UB3A expression in a number of brain regions within the rodent models. We then measured UB3A expression and compared it to rodents that were not treated with PTC-AS. Now as you can see in the figure on the right, rodents treated with PTC-AS, shown in the blue bars, had much higher levels of UB3A compared with rodents that were not treated. Now, it was encouraging for us to see evidence of UB3A expression in the rodents. However, we then had to answer the question, how does PTC-AS treatment impact aspects of the disease in the AS mice and rats? Now on the left, we conducted studies that show evidence of increased synaptic communication in the hippocampus in rodents that received PTC-AS. Now we know that rodents that were not treated showed major defects. So this was evidence of disease activity or evidence of disease improvement. On the right, we then conducted studies that showed improvement in hind limb clasping, and that's a key measure of motor function in treated Angelman rats. We also showed improvements in motor memory on the bottom right there in AS mice receiving PTC-AS. And all this was very encouraging. We then conducted some initial work in dosing monkeys, who of course have larger brains compared to rodents, have a central nervous system very similar to humans. Now in this study, we studied 2 different dose levels of PTCAS. In the monkey, we administered PTCAS into the ventricles of the brain, and we used a route of administration called intracerebroventricular route of administration. Now, the cerebrospinal fluid contained in the ventricles, once the product or PTCAS is administered to the ventricles, mixes with the cerebrospinal fluid, and then the CSF allows then the gene therapy to circulate throughout the brain and the spinal cord, which is what we want to see. Now in this initial study, we saw good evidence that PTCAS was delivered broadly throughout the different parts of the CNS using this particular route of administration. So to summarize where we are today with our gene therapy program, we've identified a lead gene therapy candidate and have conducted initial preclinical studies, and I've shared some initial data from those studies. And this year we'll be performing additional preclinical studies to generate the information needed to ultimately in the future support us to move into clinical trials. We're also establishing our internal manufacturing processes needed to manufacture the gene therapy program, needed both for our late preclinical stage and also, of course, our clinical program for dosing in humans. And we're actively involved and participating in the Angelman Biomarker and Outcome Measures Alliance to help us with planning our future clinical trials. Another important aspect is we're gaining insights from Angelman natural history data. Basically, natural history is how the disease changes over time. To inform the design of our clinical development program and allow us to put plans in place to conduct what we are planning to conduct a global natural history study in Angelman syndrome. And I'd like to tell you a little bit about that. Now, as understanding of natural history of a disease is so important, particularly in rare disease drug development, we're now in the early stages of planning a global natural history study in Angelman syndrome. So why are we considering conducting a natural history study? Well, the understanding of short and long-term disease changes in Angelman syndrome is quite limited. And it's been acknowledged that additional work needs to be done to explore potential new outcome measures in Angelman syndromes. Outcome measures meaning those key assessments that could be used in future clinical trials of your drug. Now, this study that we're planning has a number of objectives, including the evaluation of potential new outcome measures or endpoints, providing additional understanding of currently used outcome measures. We want to explore biomarkers or specific assessments that may be used to show early signs of response to an investigational treatment. Of note, data generated from this natural history study may have the potential to be used as a comparator, now what we call external control, to determine if an investigational therapy may have a treatment effect. Now, we envision this to be a global study with sites in Europe, US, and potentially other regions. This study will initially be conducted in pediatric patients, and patients will come in for regular visits and assessments to a clinic, to multiple clinics as they're involved. And there will be virtual assessments as well as in-person clinic assessments as part of the study. Now, we're in the very early stages of planning, so we'll be providing much more detail in the future about this study. We look forward to keeping you updated on PTC's gene therapy program. And with that, thank you for your time, and I'd be happy to answer questions. Thank you.
Welcome, everyone, and thank you so much for being with us today to hear from our industry partner at Biogen about their ASCSF collection study. My name's Amanda Moore, and I'm the CEO of the Angelman Syndrome Foundation, and I'm so happy that you guys have joined us today to hear more about the importance of this study. I'm joined with 2 of my friends from Biogen, and please forgive me if I pronounce your names wrong, but Rob Komorowski and Andrew Traub-Childs is with us from Biogen. So I know you don't want to hear from me and you're anxious to hear more about the study, so I'm going to pass it off to Rob.
Thanks so much, Amanda, and welcome everyone. Thanks to the Angelman syndrome community for attending today's webinar, and thank you for your interest in learning more about our CSF collection study. We're really pleased to have the opportunity to share this information with you, and during the time together we'll make introductions and talk a bit about Biogen and our research around Angelman syndrome, and then we'll dive into details related to the Angelman syndrome CSF collection study and end with a Q&A. So let's get started. So to begin, my name is Rob Komorowski. I'm a senior clinical scientist here at Biogen, and I've had the pleasure of supporting this program for almost 4 years now, and feel really lucky to have become part of this really vibrant community. And today I'm joined by my colleague, Andrew Traub-Childs, who is the study lead for the CSF collection study, and he'll be sharing the study details with you. So in case you're not familiar, Biogen is a global biotechnology company. It's based in Cambridge, Massachusetts. And at Biogen, our mission is clear. We are pioneers in neuroscience. Biogen discovers, develops, and delivers worldwide innovative therapies for people living with serious neurological and neurodegenerative diseases, as well as related therapeutic adjacencies. Today, Biogen has the leading portfolio of medicines to treat multiple sclerosis. It has introduced the first approved treatment for spinal muscular atrophy, commercializes biosimilars of advanced biologics, and is focused on advancing research programs in multiple sclerosis and neuroimmunology, Alzheimer's disease and dementia, neuromuscular disorders, movement disorders, ophthalmology, neuropsychiatry, immunology, acute neurology, and neuropathic pain. So as you may know, Biogen, Ionis, and Roche have formed a collaborative research partnership. And one of the studies being conducted through this collaboration is called the ASCSF collection study, and that's being led by Biogen. This is a study we'll discuss in detail during today's webinar. There's another study conducted as part of this partnership called the Freesia study, and that's being led by Roche. These two studies do not involve any drug. Their purpose is to help design possible drug trials for Angelman syndrome in the future. And our companies are working together on these two studies in collaboration with researchers and the families of the Angelman community so that important scientific information on Angelman syndrome is collected and shared. In parallel to these non-drug studies, our companies continue to independently research potential drug candidates for future clinical trials. So the purpose of the ASCSF collection study is to see if the UBE3A protein can be used as a biomarker for Angelman syndrome, and as well if there are other biomarkers for the disease. Biomarkers are characteristics or substances in the body that you can measure. They tell us how a body is doing, or how a body changes as a result of disease, or how the body responds to a treatment potentially. They are critical to drug development because they help us select the dose of the drug to test in a trial. They can help us to monitor the safety of the drug when it's being tested in a clinical trial. And they tell us that the drug is doing what we think it will do and working in the way we think it will work. Angelman syndrome occurs when there's a problem in the gene UBE3A, and this results in low amounts of UBE3A being made in some brain cells. The reason this is problematic is because UBE3A is needed for correct development of the nervous system, including the brain. So as a result, UBE3A protein may provide a biomarker for Angelman syndrome, but it hasn't ever been measured in the CSF of patients living with Angelman syndrome. Measuring these UBE3A levels, as well as other potential biomarkers for Angelman syndrome in the CSF, will give us an important information into the disease and what can be measured in future drug clinical trials. Because of the involvement of UBE3 protein in Angelman syndrome, we're conducting this CSF collection study. And so now I'm going to hand it over to my colleague, Andrew, to tell you more. Andrew?
Thanks, Rob. This CSF collection study was launched back in December of 2019, and enrollment is expected to close at the end of this year in August. The study is being conducted at 5 centres across the US and is looking to enrol approximately 20 Angelman syndrome participants. Unfortunately, enrolment into this study was heavily impacted by the COVID-19 pandemic in 2020. As of today, we've enrolled 4 participants into the CSF collection study, but are still looking for an additional 16. To be eligible to participate in the study, the person you represent must meet all entry criteria. The main 3 are presented here. Firstly, they must have a genetically confirmed Angelman syndrome diagnosis. Secondly, they must be 50 years old or younger. And thirdly, and most importantly, have a planned procedure that will require the use of general anesthesia or conscious sedation. This procedure must be unrelated to this study and could include, but is not limited to, procedures such as dentistry, GI tube fitting and MRIs. It is not possible to enrol the person you represent without this. If the person you represent is scheduled for a procedure requiring sedation at one of the study hospitals, you will be contacted by the study staff who will explain more detail about the study, what it entails, and ask whether you would like the person you represent to be considered for enrolment. If the study is of interest to you, you will be provided with more information about the study in the form of an informed consent form. Upon reading the informed consent form, if you would like the person you represent to be considered further for the study, you will be asked to sign the informed consent form. Prior to signing the informed consent form, if you have any questions about the study, you will have the opportunity to discuss these with the study doctor. If you sign the consent form, the person you represent will enter a screening period that may last for about 30 days. During this time, the study doctor will talk to you about the health history of the person you represent. The study doctor will also conduct a physical exam and do some tests to confirm that the person you represent is eligible for the study. If the study is suitable for the person you represent, they will have a sampling visit after all the screening investments have been completed and they are confirmed to be eligible. This can occur on the same day, but not more than 30 days after the screening begins. During the sampling visit, the person you represent will have a cerebral spinal fluid sample collected through a procedure known as a lumbar puncture. They will also have a blood sample taken from his or her vein for biomarker and DNA assessments. The lumbar puncture will be carried out while the person you represent is under general anesthesia that is required for the procedure that you already have scheduled. The lumbar puncture should not take longer than 30 minutes, but the person you represent may need to continue to lie down for 60 minutes after the procedure to reduce the likelihood of any discomfort. You and the person you represent will also need to be available for a follow-up contact about 72 hours or 3 days after the sampling visit. The person you represent will usually be discharged following the lumbar puncture once the hospital has confirmed it is safe to do so. The follow-up contact will generally then be conducted over the telephone once you and the person you represent are home. As safety is our highest priority, study participants will be assessed and monitored at the study site and during the follow-up contact for any side effects. The study will not directly help the participant, but he or she will help researchers improve their understanding of Angelman syndrome and help find potential biomarkers for use in Angelman syndrome clinical trials in the future. When a study participant joins a CSF collection study, you will have the opportunity to opt in to place them on a priority screening list. The priority screening list may give the participant the opportunity to screen for a planned investigational study in the future. However, this does not guarantee enrolment, as several requirements would need to be fulfilled prior to enrolling in an interventional trial. We are very grateful to all families who volunteer to take part in research and drug development efforts. As discussed in an earlier slide, there are 5 study hospitals that are conducting the CSF collection study as outlined on this slide. If you are interested in learning more about the study and possible involvement for the person you represent, please contact the location that is most convenient to you. Thank you for your time and attention. We can now open it up to questions.
Thank you so much. That was such wonderful information. I think that both this study and the Freezer study is such a crucial study to support biomarkers and endpoints and understanding Angelman syndrome as we move forward and as we get into this really exciting period of having phase 1 trials. Like this is so exciting for our community. So I just have a few questions to ask you guys. As we move forward here. I think we talked a little bit about the actual procedure and the actual procedure will be about 30 minutes. How long, you talked a little bit about this, but how long will the child have to remain under anesthesia after procedure? Because being under anesthesia is sometimes a concern for our community. So just understanding a little bit more of how long they will be under anesthesia after the actual, collection of the CSF sample.
Yeah, sure, Amanda. I can take that. So the spinal tap itself should prolong the anesthesia or sedation for about 30 minutes. So relatively 30 minutes after the procedure. Yeah.
So and then to put some ease to mind of families, because when we talk about these procedures, when you talk about lumbar punctures, especially for families who have never experienced this before, it can be a really scary thing. So can you talk a little bit to me, either Andrew or Rob or one of you? One, is it safe? And what kind of side effects can we expect, or the child expect, or the individual with AS expect from this?
So I can take that one, Amanda. So spinal tap or lumbar puncture is a standard procedure used in medical practice. There are potential risks associated with spinal tap, and those possible side effects of the spinal tap procedure are generally headache that may or may not be accompanied by nausea, vomiting, dizziness, and ringing in the ears. Is caused by the leak of the cerebral spinal fluid after the spinal tap. Also low back discomfort or pain, and then infection and bleeding at the site of the spinal tap.
So I'm curious, because a lot of the concerns our families have, with having individuals with Angelman syndrome being nonverbal, how will we be able to tell if there are side effects happening with our children? I mean, outside of the actual vomiting, if they're having headaches, like what if they can't tell me? What's the process there?
Yeah, so the guidance we have is that you should monitor him or her and rely on the cues that indicate that they're just not feeling like their usual self.
You talked a little bit about the safety follow-up, but how is that conducted, and do they have to come back to the actual hospital site for that?
The safety follow-up visit is conducted approximately 72 hours rather after the LP procedure has taken place, when the person you represent has been discharged and is back at home. The study aim is to conduct this visit over the phone. Wonderful. And it is an opportunity for your doctor, your study doctor or designee to check the health of the person you represent. Your study doctor will ask whether the person you represent has had any side effects after they left the hospital following the procedure, and whether any of these side effects are still ongoing. In the rare situation where the person you represent is required to remain in hospital for more than 72 hours following the pre-scheduled procedure and the LP, then the safety follow-up visit will be conducted in person by your study doctor or designee. The hospital stay may be extended for monitoring purposes, which may be a requirement of the pre-scheduled procedure, or may be due to complications resulting from the pre-scheduled procedure or the LP.
So the good thing is, in most cases, they won't have to travel, because our families, when it comes to traveling and going back, it's sometimes not fun. So that's good to know that in most cases, hopefully that can be conducted over the phone. So Rob, let's talk a little bit about the samples. So what will happen to the leftover samples? Are they stored, and for how long? Do I have control over how those persons' samples, how they're being used?
Yeah, sure. So all the samples will be used for the purpose of the study are outlined in the consent form. It's a bit difficult to determine whether there will be any leftover sample, and this really will depend on the volume of the sample collected during the study and the extent of the sample analysis required to meet our study goals. So in the event that there is residual or leftover samples, you'll have the option to consent to having these samples used for future scientific research, and this consent is optional. So if you decide not to opt in, this won't prevent the person you represent from taking part in the main study. So any residual samples to be used for that future scientific research will be stored for as long as it's required, or a duration that's dictated by the local, national, or regional laws and regulations. Additionally, as part of the study, a DNA sample will be collected and analyzed, and this will be retained by the sponsor for 25 years, or again, a duration that's dictated by the local, national, regional laws and regulations.
Okay, Andrew, so if I participate at one location for the study, can I apply? So we're going to talk a little bit, I should frame this up. We're going to talk a little bit about the priority screening. Priority screening, you spoke a little bit about this, that if an individual participates in the study, that they can, get this kind of a priority to be screened, which is different than actually being enrolled into the study. So just being screened to see if they meet all their criteria to move on on to that next step. So if I participate at one location for the CSF location, study, can my priority screening be applied at a different location for whatever, for that next study?
Yes, absolutely. All future study locations will honor the priority screening status.
Perfect. That's a simple answer. I love it. So one more question about priority screening. How will the person I represent's priority screening status be honored, Rob?
Yeah, so when you join the CSF collection study, you'll have the opportunity to opt in to place the person you represent on the priority screening list. And this will be documented in the participant study notes. So the study team will be aware which patients are eligible for priority screening at the start of any interventional trial. IQVIA is helping Biogen conduct the study, and they'll keep a full list of the patients that are eligible for priority screening across all the sites. So Biogen will not have any access to patient identifiers. The tracker will also detail whether the priority screening will take place at the hospital site where the patient is enrolled in the CSF collection study, or whether this will take place at an alternative hospital.
Okay, perfect. Andrew, if I don't live near a study site, but the person I represent is having a procedure which requires anesthesia, can I still participate?
Yes, yeah, again. So as long as your insurance covers the pre-scheduled procedure to be carried out at one of the study sites, and the person you represent meets all eligibility criteria for the study, then they will be able to participate. Absolutely.
Perfect. So I want to just so people kind of understand why some of this is necessary, Rob, when we talk about spinal fluid and, the understanding the worth of that, why is spinal fluid necessary? Couldn't you just take a blood sample from this?
Yeah, it's a great question, Amanda. Cerebral spinal fluid, or as we're referring to it, CSF, can provide important information about Angelman syndrome that really can't be seen in the blood or urine or saliva for that matter. So by collecting a sample of spinal fluid, we'll be able to learn more about Angelman syndrome and be able to better test for potential biomarkers.
I have one more question for you guys. I hope it's kind of inundating you with questions today. So, Andrew, I'm someone sitting at home anxious to possibly do this study. If I represent, if the person I represent is not scheduled for a procedure, can he or she still take part? How does that work?
No, unfortunately not. A prerequisite of the study is that the participants have to be attending the study site for an already pre-scheduled procedure that requires anesthesia or conscious sedation. The reason for this is to minimize participant trauma, really, and upset, and then also additionally any burden to caregivers, parents, by having to make an additional visit to the hospital purely for the study.
And we appreciate that, because that caregiver burden with travel and other ways can be really hard. So I think there's probably a lot more questions I can ask, but I appreciate your time today, understanding the study and getting it out there. COVID has been hard on us in so many ways, but definitely it was hard on this study. So, for all the families that are out there listening right now that are interested in this and want to understand more or learn more, one, you can reach out to the foundation. We're more than happy to answer any questions that you may have. Go to clinicaltrials.gov. That's always a great resource to understand about the study, about the research. We also have Angelman Clinical Trials website that has all the information as well. So there's multiple different you can go to learn more about the study. Also, you can call the PIs at the locations and just talk to them. Have a conversation. You're interested in the study. I want to learn more. But this is a really necessary thing for our community, and we've got to have this information in order for us to move forward. So, Rob and Andrew, thank you so much. I want to thank Biogen for the commitment they have to the Angelman syndrome community. It means so much to us, and we're just so thankful for the work you're doing, and we're looking forward to what's come. And I just encourage families that if you're interested in participating, please do. So thank you, guys. Hope you have a wonderful day, and we'll talk with you all soon.
Okay. Thanks, Amanda.
Thanks a lot, Amanda.
Thank you.
Bye.
Thank you for your interest in the Angelman syndrome natural history study. You have now heard a lot about the very exciting and promising clinical trials that are coming down the pipe that will probably change the face of Angelman syndrome. I'm going to tell you today why it is really important to take part in the natural history study and the role that we play in drug development. So why should anyone participate in the natural history study? Well, it turns out that the natural history study is really important for development of therapeutic products, as many leaders of the field in the field of drug development have actually stated in the past. So the natural history study allows us to determine whether a compound that is being trialed has specific side effects that's unique to the compound, or whether it is just part of Angelman syndrome. It also tells the drug companies the endpoints that can be useful in clinical trials. And as has also been said, having a rigorous natural history study is really important for generating the kinds of data that drug companies can use to design their study and to determine the outcome measures that they should use. So the natural history study in Angelman syndrome really started back in 2006. That was the first time we had ever conducted a natural history study. And at that time, we enrolled all participants with Angelman syndrome, and they came to see us once a year. The only real criteria is that they had to have a molecular confirmation of Angelman syndrome, and they should not have any other condition that contributes to symptoms that could mimic Angelman syndrome. We enrolled 302 subjects across all sites. And one of the outcomes of this study was really the development of EEG as a biomarker in Angelman clinical trials. So as you can see, many researchers have used our data both in academia and in industry to further develop EEG as an outcome measure. And it is because of all these studies using the data that we generated from the first Angelman syndrome clinical trial that we are now able to use EEG as an outcome measure in all the current clinical trials. Because this is a longitudinal study and we have been seeing participants in our study once a year, we have been able to generate longitudinal data to understand how changes in specific components and aspects of Angelman syndrome change over time. So for example, we were able to show in this study that anxiety increases over time. We would not have been able to do this if we had not collected data longitudinally from multiple visits. As you can see, these are the 302 participants in our study. And we were heavily biased towards the younger kids. We had very, very few adults in our study. This is a problem because it basically means that we have not been able to learn enough about the complications and manifestations of Angelman syndrome in the adult population, which limits our ability to help all of you who have adult children with Angelman syndrome. You will also see that we had quite a high dropout rate, that even though we had 302 participants who came for the initial visit, very few of them sort of came for follow-up visits. So we had over 200 participants who came for 2 visits, about 160 who came for 3 visits, and very few came beyond that. So this again limited our ability to study Angelman syndrome in a longitudinal way to help people understand, to help all of you understand how the various symptoms change over time. So when we were refunded by the FDA to conduct the study in 2017, we made a few major changes to our study design to try to address some of these limitations. First of all, we consulted the various stakeholders in the selection of our outcome measures. So we talked to members of the ABOM, We talked to Angelman Syndrome Foundation and the Foundation for Angelman Syndrome Therapeutics, and we talked to various companies that were developing therapies for Angelman syndrome, and we asked them, what outcome measures would you like to see us investigate in this study that would help you develop new therapies for Angelman syndrome? We recognize that all of you invested a lot of time and energy coming to these visits, and we have been very grateful to all of you for the effort that you have put in to help us with our first natural history study. So when we developed this study, we wanted to try to find ways to minimize the burden that participating in such a study would cause to you. So one of the features in this new study is having an online portal that you can complete in your own time at home, as long as you have an internet connection, and you can save time by not having to complete these forms on paper on the day of the visit. Now, for those of you who would prefer to complete these forms on the day of the visit or on paper, that remains an option, but we do want to emphasize that this online patient-facing portal should really help streamline the visit and save a lot of time. The other innovation we have developed through this study is what we call a hybrid model. So what we are doing now is to complete part of the visit online. So you will meet with the study staff, a coordinator, the investigator, over Zoom or some form of telemedicine platform. You will complete all the questionnaires online in your own home or your own environment. And then you come to the study site just for an in-person assessment that will be much shorter. So instead of spending 5 or 6 hours at a site, now the total amount of time that we would need you at a site will be reduced to about 2 hours. Our hope is that that again will help to make it easier for you to participate in this study. In our new study, we really place a lot of emphasis on the outcome measures that will be used in the future clinical trials and indeed in some of the current trials that are ongoing. So you're all familiar with the Bayley scales of infant and toddler development. We recognize that this is not the best scale to be used for in the Angelman population, but unfortunately, it really is the outcome measure that the FDA is particularly interested in and focused on. Therefore, you will find that most clinical trials are actually using the Bayley as a primary outcome measure. Now, what happened was that a new edition of the Bayley was released in September of 2019. And so we have very little data on the Bayley, the 4th edition of the Bayley in the Angelman population, because soon after this edition was released and after we incorporated this into our study, unfortunately, COVID hit and we were not able to conduct in-person assessments. We cannot complete the Bayley without an in-person assessment, so we really, really need these data for the future clinical trials. We also started administering the 3rd edition of the Vineland really from the inception of this new study. And again, this is important because in the previous study, we were using a 2nd edition because that was what was available at that time. But the new clinical trials now are all using the Vineland 3rd edition, and we therefore need data from that. As some of you are aware, the ORCA was developed through a collaboration with our colleagues at Duke University, and they released that in 2020. So we are now starting to collect data using the ORCA. I should also mention that we will be conducting an off-site visit, study visit in Princeton, New Jersey in September of this year. So we are hoping that some of you will be interested in coming out to Princeton, New Jersey to see us so that we can perform the Bailey, as well as other in-person assessments with you at that time. That would save you a visit to your local study sites. And if you happen not to live close to any of the other local study sites, but are interested in participating in this study, this may be a great opportunity for you to participate in our study and to contribute to the development of clinical trials. Again, we are very conscious of how much time and effort this imposes on you, and we are really grateful to all of you for your participation. We really could not have done this without you, but I think it is important to really recognize that every parent has a role to play in helping us to bring therapies, effective therapies, to the Angelman syndrome community. So we have currently enrolled across all study sites in the USA and Canada, 159 participants with ages ranging from 5 months to 52 years. But as you can see, we are still heavily biased towards the younger population. So we certainly have had more adults now in this study than we did in the previous study. So I think things have improved, but over 80% of the participants in our study are still under the age of 20. So again, I'm appealing to all of you who have adults with Angelman syndrome to please sign up for this study, because we really need to learn from you in order to help you manage your children more effectively. So these are all the study sites in our study. So as you can see, there are 7 study sites across the country in the USA, and we actually have 3 study sites in Canada. So for those of you who live near Canada and COVID permitting, if you are willing and interested in going to Canada, you can certainly join the study through the Canadian sites as well. So if you're interested, please contact Jonna O'Sullivan, who is the main coordinator for this study. So you can see Jonna here, and that's the email address for the study, as well as Jonna's email and telephone number. We also have 2 new coordinators who joined our team this year, Katie Anderson on the left and Liana Ko on the right, and you may hear from one of them as well. And finally, again, just want to thank all of you for the many years that you have supported us through this study. Thank the Angelman Syndrome Foundation and Foundation for Angelman Syndrome Therapeutics for their continuing support. We are also collaborating with RTI International to analyze a lot of our data, and they have been great to work with. And finally here, these are the funding sources that we have received over the years. So thank you for your attention, and hope you will participate in the study. And I just want to take this opportunity now to introduce you to our wonderful team of study staff at Boston Children's Hospital. So you can see Katie Anderson, Jonna O'Sullivan, and Liana Ko. Thank you.
What an amazing night. That was a lot of information, and a lot of you have asked if we've recorded the session, and we have recorded it. So it will be offered and up for you guys to review afterwards. But I don't know about you, but my heart is leaping for joy. It is so amazing to see so many people in the field working so hard for our individuals with Angelman syndrome. And I am so hopeful, and so should you be. So thank you guys for keeping it hanging in with us tonight. It's been a long 2 hours, but hopefully it just gave you all the energy and excitement as we move forward. So I just want to thank you all and get you excited for tomorrow night. Every night this week, we will have an awesome panel for you. Tomorrow night, it's all about clinical trials. So come hear all about the different clinical trials, how to prepare, what's going on, all that fun information. And it will start at 8 p.m. Eastern. We're hoping you're enjoying the conference so far, and we love you all, and we'll see you tomorrow.